Hello, welcome everyone to the Arthritis National Research Foundation's Researcher Spotlight webinar series. My name is Ashley Barrow. I'm the Director of Individual Giving here at the ANRF. And on behalf of our Board of Directors, Scientific Advisory Board and staff, I would like to welcome you all to the Spotlight on Osteoarthritis. We're so excited to have so many of you joining us today. We would also like to thank our corporate partners for supporting the Research Spotlight series. A very special thank you to AbbVie for being our gold level sponsor and for your continued support and dedication. It's because of these partnerships that we're able to continue to pave the way for new innovations and discoveries from our researchers. So before we get started, there are a couple of logistical pieces of information that we want to share. First is that the webinar is being recorded. We will have it available on our website and YouTube channel in the coming weeks. Second, if you have questions during the webinar, please type them into the Q&A. If we aren't able to get to yours, please email info@curearthritis.org after the webinar with your question, and we will get back to you. Now, without further ado, I'm honored to turn the mic over to our patient moderator and ANRF board member, Staci Stringer, to tell us her story of living with OA. Thanks, Staci.
Thank you, Ashley. Again, my name is Staci Stringer, and I'll be your moderator today. I am a member of ANRF's Board of Directors and also patient advocate. I live in Southern Oregon and have been living with rheumatoid and psoriatic arthritis for 19 years. And recently, in the past 2 years, I added the diagnosis of osteoarthritis. I was diagnosed at the age of 18 and had to give up my volleyball scholarship because none of the doctors knew what was wrong with me at the time. No one considered that I could have arthritis at 19 because I was too young. I struggled for 15 years to find a medication that worked well. I've jumped from therapy to therapy every 2 years or so in search of feeling normal. After struggling to find doctors to fight for me, I traveled to Johns Hopkins Rheumatology Clinic in 2021, and now of a physician, that has been fighting for me and stepping outside of the box with my different treatment options. As I've been running out of options because I've jumped through too many medications. So this is part of why ANRF means so much to me, is that the research that our scholars are conducting will directly affect my quality of life and my future. So I'm extremely invested in the work that they are doing and I'm excited to continue the research discussion today. So enough about me. I'd love to pass it over to our first presenter. Dr. Matlock Jeffries is a leading osteoarthritis researcher and clinician at the Oklahoma Medical Research Foundation and the VA. His work focuses on understanding how factors beyond genetics, like gut bacteria and cellular changes, affect knee osteoarthritis. Dr. Jeffries' team was the first to study bacterial DNA in joint tissue and explore how gut bacteria might influence osteoarthritis differently in males and females. He also developed a blood test to help predict how OA may progress in patients. Dr. Jeffries has received funding from major health organizations, and he was recognized as an ANRF scholar in 2017. I'll pass it over to you, Dr. Jeffries.
Great. Thanks so much, Staci. I really appreciate the opportunity to come and give a little bit of our research. So sorry, the slides are just a touch delayed. A bit more about me. I'm a native Oklahoman. I'm from a small town in northeast Oklahoma that you can see on the map here. I went to both of our public schools in the state actually for undergraduate. I ended up graduating from OU down in Norman. I worked as a research technician before I went to medical school for a year, and that's really what stimulated my desire to become a physician scientist and to do more scientific work. And then I did all of my sort of standard training, internal medicine residency and chief residency, and then finally rheumatology fellowship, which I graduated in 2016. And I've been on faculty since then, originally at the university and now here at OMRF. And I also have an adjunct appointment at the VA where I see a number of patients, both that have autoimmune forms of arthritis, as well as those that have osteoarthritis, which is, of course, the disease that I study. So for those of you that aren't familiar with OA, OA is the form of arthritis that most people mean when they say that they have arthritis. It's by far the largest number of patients that have arthritis have OA. It's the leading cause of chronic disability in the U.S. More than 20 million individuals, probably quite a bit more than that, we just don't have great records, are affected by OA. It costs the economy at least about 1% of GDP when you factor in work loss associated with arthritis. During fiscal year 2011, which is the last time we have really good numbers for this, the Medicare reimbursement for OA joint replacement, specifically knee joint replacement, was the largest single line expenditure in Medicare's budget. And OA continues to be the leading cause of medical discharge from active military duty. Much of this is post-traumatic OA related to injuries. We do understand these days that OA is a disease of chronic, low-level, all-over-the-body inflammation. There are always, of course, components of malalignment or abnormal joint loading or damage, but it's not a disease of wear and tear. That's far too simplistic of a way to think about OA. It's strongly associated with the part of the immune system we call the innate immune system, both within the joint and all over the body. There are a number of factors that are associated with OA development, and these include age and obesity and sex. There's a slight female predominance to OA. Ethnicity as well. It appears that African American and Native American patients are at increased risk for OA compared to white folks. A history of previous trauma increases your risk of OA, and then there is certainly some component of genetic risk, and a decent amount of OA is heritable, although it's interesting, the heritability changes according to the joint that you're talking about, with knee OA being amongst the lowest risk and hand OA being amongst the highest risk from a heritability standpoint. Something else that is really interesting, and I show this slide every time I give a presentation, a lot of people don't realize this, is it does appear based on the scientific literature that the rates of knee osteoarthritis have increased dramatically since industrialization. So there was a really nice paper published, about a decade ago or so, that went back and looked at some of our historic counterparts in the prehistoric era and some skeletons from the early industrial era, and showed that we have a rough doubling of knee OA prevalence, both among men and women, that started right around the time of industrialization. And importantly, this isn't just because we're living longer and that we tend to be heavier. We see these increases throughout the age spectrum, and even when you control for rates of obesity, this still comes out. So this is an important problem that needs to be tackled, I think. So something else that really stood out to me whenever I was doing my clinical training, and the reason that I shifted my research focus from lupus, which is what it was back at the beginning, to osteoarthritis here later, is that we really don't have any good drugs to treat OA. You know, in my regular rheumatology clinic, I treat patients that have autoimmune arthritis like rheumatoid arthritis, which is about 1% of the population. And we have countless drugs that are disease modifying, 20 plus drugs that are disease modifying for that disease. But for OA, that is, 20 to 40 times as common, we don't have anything that is FDA approved that will actually modify the course of the disease. So this is a short list of drugs that we use to treat rheumatoid arthritis, and every single one of these can prevent the disease from progressing. But for osteoarthritis, that's more than 20% of the population, we don't have anything. So my laboratories worked for a long time to try to figure out what it is that is causing osteoarthritis, why we're seeing this increased risk of osteoarthritis over the last 80 to 100 years. And one environmental factor that we sort of stumbled on, but we think is really interesting, is the influence of the microbiome. So whenever you say the microbiome, generally what people are referring to are bacteria that live within the gut. And our laboratory decided to take a slightly different approach and look for traces of bacteria in the places where we get arthritis within joints. So the first thing that we did and published here a number of years ago is we just looked for bacterial DNA within the joints of patients that were undergoing joint replacement, both in knee patients and hip patients. And we found a couple of things that was intriguing. The first thing that we found is that there were bacterial DNA in every sample that we looked at. And we sort of didn't believe it at the beginning, but then we went back and collected a very large number of these samples in a sterile fashion and found that they really were there. The other thing that we found, and what's illustrated in the graph on the right, is there are a number of bacterial species that are significantly different in OA cartilage compared to patients that don't have OA. We had gotten cadaveric controls for this particular experiment. So we're now in the process of trying to figure out what this has to do with OA, if it's just along for the ride, or if it has something actually to do with disease pathogenesis. We'll be able to answer a few of these questions, but we're still working on the rest. So the first question that we asked was, how does it get there? It appears, based on our mouse models, probably does come from the gut. We see that we start seeing joint bacterial DNA pop up about 48 hours after you give a mouse that doesn't have any bacteria at all, an oral bacterial load. And then we wonder, well, can you change it if it's associated with OA? Is there some way to alter the pattern of the microbial DNA within joints? And the answer is yes, you can, at least in animal models. It takes some time, but you can modify it. Are these patterns associated with OA risk factors? And we found that both aging and obesity so far have been associated. And then the final question is, does this bacterial DNA actually have anything to do with OA? The jury's still out on that, but I think the answer is going to be probably so. So I'll show you really briefly in the next slide some example knee histology from a mouse. This is just one half of a mouse knee. Everything in red on this histology slide is the cartilage, and the blue is the bone and other connective tissue that's around the joint. We did this experiment where we took, again, these germ-free mice that don't have any bacteria at all, and then injected their joints either with saline or some control DNA or some bacterial DNA that we got out of actual human OA patients. And what we found is that when you inject bacterial DNA from human OA patients, we see a really significant increase in the damage that's associated that we see in mice that have OA, and that's in this bottom panel. So we think there may actually be something going on here. The final little tidbit to this is, what does this have to do and how is this working? And the answer is we don't know yet, but it's interesting. There's some other people that have published various mathematical formulas to try to figure out how sort of inflammatory DNA from bacteria are when it's exposed, when your host immune system is exposed to it. And what we find when we run the sequences for the bacterial DNA we find in OA patients is it does appear to be significantly more inflammatory or expected to be more inflammatory than patients that don't have OA. So I think the answer is that the DNA that is within our joints that's associated with bacteria might be more inflammatory in patients that have OA compared to patients that don't. Still a lot of work to go in this particular area of research, but we're getting there. The other thing that my laboratories looked at, which is sort of related to this, is well, we know that the microbiome, especially the gut microbiome, can be modified by diet and by probiotics and things like that. Might this be useful as a treatment for osteoarthritis? To do an evaluation of this, we of course focus on mice, and it's interesting. The regular mice that we used for osteoarthritis research are the mice that you see me holding on the left here, the black or brown mice. And they get osteoarthritis as you would expect whenever you do a certain surgery to their knee. But there are mice that for whatever reason don't get OA whenever you would do the normal surgical interventions that would ordinarily induce it. An example of that mouse is the white mouse on the right side. And these mice are really interesting. These are called MRL mice, and they do a whole bunch of things that you might consider extraordinary from a tissue regeneration or healing standpoint. So one of the things that they do is they can heal certain size skin wounds. So for example, you can see on the mouse, the brown mouse on the left, he has a little hole punched out of his ear, right? This is a certain size of ear hole punch we use for identification. And the white mouse on the right received a punch at the same time, but he's almost completely closed it. These white mice also can heal skeletal muscle to a certain degree, repair tendons to a certain degree. And it hadn't really been worked out exactly how this was working. And one of our hypotheses was, well, maybe the gut microbiome has something to do with this. So what we did is we basically did reverse microbiome transplants. So we took the microbiome from the healer mouse and put it into the regular mouse and vice versa. And lo and behold, what we see, which is on the left panel of this of the graph here, is that when you transplant in an MRL or a healer microbiome into a regular mouse, you significantly improve their knee osteoarthritis risk, basically taking them down to where they get very, very minimal osteoarthritis. Conversely, you can actually make the healer mice worse by putting a regular mouse, which is denoted here as B6 or black 6, into them. They don't worsen quite as bad as the black 6 regular mice improve, but you can still make them worse. And some other stuff that we found that was really intriguing is on this next slide, which hopefully will pull up for us, which is that these differences in osteoarthritis risk based on the microbiome are actually heritable. So if you take the transplanted mice and breed them together, their children generation, here we go, also are protected from osteoarthritis, and then their grandchildren generation are still mildly protected from osteoarthritis. And I think it sort of slowly goes away over the different generations. And we do see improvements in inflammation within the joint, as well as extra bone formation, which are called synovitis and osteophytes here. And I have a few examples to show you too, because I don't know about you, but I'm sort of a visual learner. So on the left-hand side here is a similar view of a mouse knee. This one has osteoarthritis. You can see that red is thinned and it's got a very rough texture to it. On the right-hand side here is one of these same mice that we transplanted in the microbiome from one of these healer mice. See, there's very minimal osteoarthritis here. And then the next page, we see sort of a similar thing. On the left is one of the healer mice by itself. You can see they really don't get osteoarthritis. And on the right is a healer mouse that got one of the worse microbiomes. You can see we're starting to see a lot of fissuring and such there. So this is very, very interesting to us, and we're following up on this now. Still don't know exactly how this is working, probably by modulating inflammation, but we're working on that. Something else that we found that was curious is very specific species within the microbiome strongly correlated in these mice with whether they got OA or didn't. One of the most intriguing species that appears to be correlated with protection against arthritis is this genus called Lactobacillus. And you see Lactobacillus a lot in various probiotics and things like that. There's actually some probiotics that have been shown in studies from Japan that included large amounts of these Lactobacillus, that they may be beneficial in patients that have knee osteoarthritis from a pain perspective. So I think that's really intriguing, and we're following up on these now. So in summary, we think that both the gut and cartilage microbiome probably have something to do with OA. The cartilage microbiome manipulation appears to perhaps worsen OA, at least the microbial DNA component of it. We don't yet know if there are live bacteria within your joints or not, or if it's just the nucleic acid material. But either way, it appears that it worsens osteoarthritis a bit. We do know now that gut microbiome transplantation can prevent osteoarthritis, at least in mice. We're not there yet in humans. So then the big question is, how does this get applied to human patients in the clinic? Could we use microbiome modifications to treat instead of just prevent osteoarthritis? And the answer is we just don't know yet, but that, I hope, is where we're headed in the next few years. And that's my final slide. Thanks to all of the folks that have given us money in the past, and to my lab techs that work all the time to try to produce these data for us.
That was absolutely incredible, Dr. Jeffries. I learned so much from that, and I really love that you broke down showing us the different visuals of the mice knees, and actually a lot of that might be applicable to my case. So thank you so much for sharing. I'm going to pass it over to Dr. Rachel Miller. She is a researcher at Rush University Medical Center with over a decade of experience studying joint disease like osteoarthritis. She specializes in understanding how mechanical forces in our bodies contribute to pain, especially in osteoarthritis. By using advanced engineering tools and mouse models, Dr. Miller's lab is working to uncover new treatment targets. She has published extensively on musculoskeletal disease and was awarded an ANRF grant in 2017 to explore innovative ways to treat osteoarthritis. So I'll pass it over to you, Dr. Miller.
Okay, thank you so much, Staci, and thanks everybody for joining us today, and for Matlock for doing a great job in introducing our topic for today. So before I get started on my research, I'd just like to highlight that I'm a member of the Chicago Center on Musculoskeletal Pain. The director of the center is actually a board member of ANRF, Dr. Anne-Marie Malfay, and so I invite you to check out our website to learn more about what we do. And so, as Matlock introduced, osteoarthritis risk factors include things that affect a large portion of the population, which results in a lot of us eventually suffering from osteoarthritis. So whether that's aging, joint injury, or obesity, all of these things can lead to joint instability, as well as systemic inflammation, like Matlock described, which can further the development of osteoarthritis. So by X-ray, you can detect joint space narrowing, which happens when your cartilage starts eroding and your femur and tibia get closer together. But by MRI, you can appreciate other soft tissue involvement within the knee joint. So what's showing up in white here is areas of inflammation. So whether that's synovitis or bone marrow edema, all parts of the knee joint can contribute to the disease that we know as osteoarthritis. And unfortunately, there are limited treatment options. There aren't any for slowing the disease progression, but there are also limited options for managing the symptoms. And so osteoarthritis remains a leading cause of all the diseases considered of chronic pain worldwide. And so when we're trying to think about why osteoarthritis causes pain, we have to consider all aspects of the nervous system. So within the knee joint and your other joints that would be affected by osteoarthritis, there are nerve endings of sensory neurons, the nerves that sense pain. And so when there's different activators of pain in your knee joint, this causes an electrical signal to get started and propagates up along the nervous system, eventually reaching your brain, where it's interpreted emotionally as pain. And so when we're thinking about chronic pain, that leads us to try to understand what about the nervous system has changed so that it's not just an acute sensation of pain, but pain that affects your daily life. And so some terms that I'll be using throughout my talk today have to do with this concept of the nervous system changing, which we call sensitization. So basically what that means is the neurons that normally sense pain when, for instance, when you touch a hot stove, you sense that as being painful. When your nerves are sensitized, now they'll start to respond to, in the case of your stove, lower temperatures or to the same temperature with a more painful feeling associated with that. And so these two concepts of responding to lower level stimuli or more to the same level stimuli are called allodynia or hyperalgesia. And so in the lab, we have ways of studying this, and also in the clinical world, they do as well. And so what our colleagues in the clinical world have noted is that patients with osteoarthritis have pain that's often activity related, but they also show signs of this sensitization. And so in the clinic, they use devices like what is pictured here in the lower left-hand corner. It basically allows the researcher to apply a certain force to the knee joint, and then you can keep applying more force until the patient responds and tells you that it's now painful. And so in a normal person, you would get a certain number, but in patients with osteoarthritis, the force that is required to elicit pain drops. And so this is what we call a lower pain pressure threshold. And we find this interesting because these numbers can correlate with your pain severity, but it can also predict the future development of pain. And so an interesting study that came out about 5 years ago now showed just in a cohort of the general population of people with or without osteoarthritis, they would take a baseline measurement, and that baseline measurement would help the researchers predict who would go on in a few years to develop persistent knee pain. And so in the laboratory, we use mouse models like has been introduced. And so then we have come up with ways of modeling these different concepts in our mouse model. So we can induce osteoarthritis through surgeries. So we can cut ligaments inside the mouse knee so that the mice go on to develop osteoarthritis over the course of weeks to months. And we can use similar kinds of tests like I just described are used in the patient population. So in the upper picture here, you can see it's the same kind of device that's used to apply forces to the mouse knee, but this is a mouse-sized version. And so we can, from the mouse, get information back about what forces are required for the mouse to feel pain, just like we did in the patients. And this generally starts to appear early in the model and then remains present throughout the course of osteoarthritis. Whereas other types of behaviors only start to come out towards the end of the model. So like Matlock showed, when cartilage erosion starts happening, here you can again see in the right-hand part of the picture here, the cartilage is more fibrillated and you can see loss of the pink signal. And so when that happens in the animals, they also start to put less weight on the operated limb that's suffering from osteoarthritis, So here's our little assay. It might be hard to see in the video on the screen here, but we train the animals to stand on 2 force plates, and then they pull on the string, and at the end of the string is a Cheerio. So that's how we teach them how to stand on the device, and then that allows us to measure how much force the animal is putting on each leg, so that we can see that the leg with osteoarthritis, eventually they put less weight on compared to the normal leg. We can also do different kinds of neuroscience experiments to see how the functions of the nerves are changing over the course of the disease model. So the picture here is showing actually the sensory neurons, the neurons that are sensing pain, responding in real time as we apply forces to the knee joint. So the neurons will get a brighter green color, and we can quantify that and see how the nerves have changed because the mice have osteoarthritis. We can also see anatomically where the nerves are located in the knee joint, and that's what I'll be going on a little bit in the next portion of my talk. But before I get here, this is just a nice overview of some of the different projects that we focus on in the laboratory. I could add microbiome to the top of the picture here as one of the systemic or inflammatory inputs. But it's just kind of trying to capture the idea that changes are happening all over the nervous system, not only at the level of the knee joint, but also at the level where some of the connections are made along the way in your nervous system. So I have some exciting pictures to show, but I think that causes the slide advance to be a little bit slow, so bear with me here. So what we can do in the lab is actually use different types of genetically modified mice that allow us to see where the nerves are. So what these pictures are showing you in red is where the nerves that sense pain are located in the mouse knee joint. So on the top is a normal joint, and on the bottom is a joint with osteoarthritis. So you can see the blue arrows are pointing at areas where nerves have now grown in where they didn't used to be in the normal joint. So underneath where the cartilage is wearing away in the bone, that's the first arrow on the left, you can see nerves now growing up through the bone towards where the cartilage lesion is formed. And on the right, those 2 arrows are pointing at new nerves now present in the synovium or joint lining, where often we note inflammation happening in the knee joint. And so we can do this reproducibly, causing osteoarthritis a number of different ways in different mouse models, but it keeps happening. So we keep finding new nerves in the synovium and new nerves in the bone underneath where the cartilage erosions are forming. So it's of interest to try to better understand what kinds of nerves these are and what druggable targets they could be expressing, so that we could potentially develop novel pharmacologics to go after and diminish the ability of these neurons to be signaling pain. As was mentioned in my introduction, we're also interested in trying to include different kinds of engineering tools along the way. So this is a new way that we're working on now to visualize where the nerves are in the knee joint by actually taking the intact mouse knee joint, that's the first picture on the left, treating it with a different combination of chemicals to make the knee joint transparent. And then you can actually in 3D see where the nerves are instead of having to slice through the knee joint and look one section at a time. So we're pretty excited about this. This is our kind of first knee attempt here, but tomorrow we're looking forward to our next batch of imaging, so we'll see what happens. Just to say that this is also relevant in people. So this is a group from England who's started to look for nerves in the subchondral bone in patient samples, and you can see that the number of nerves that are growing up through the bone increases in patients with symptoms compared to asymptomatic controls. We're also working on this as part of a collaboration with other groups around the country through NIAM's NIH-sponsored program called Rejoin. And so for the rest of my talk, quickly, I'll be telling you a little bit about a different project where we're actually looking how the nerves respond to mechanical forces in the knee joint. And so this project I got started on through my ANRF funding back in 2017. And in 2021, the person who discovered these ion channels was awarded the Nobel Prize for his discovery, Artem Patapushin. So what these channels are You can see here, it's a pretty large protein expressed on your cell membrane, and these ion channels respond directly to mechanical forces. And so one of these, piezo2 is the name of it, is particularly expressed by neurons that sense pain. And people have studied them in different contexts. One of the earliest ones was an observation in a small group of people that genetically do not have functional piezo2 channels. And so they were trying to study what made these people different from healthy controls. So what the test was that they did is they applied capsaicin patches to their skin, waited 30 minutes, and then pressed on that patch of skin compared to a control patch of skin, and asked them, which skin did I apply the capsaicin to? So if I did that to all of you, I'm pretty sure all or most of you would be able to for sure tell which patch of skin got the capsaicin, but the people who are missing piezo2 couldn't tell the difference. So they never got sensitized to the capsaicin. So this observation got us thinking, could piezo2 also be involved in the ability of these nerves inside your knee joint to become sensitized in osteoarthritis? So we did a similar kind of experiment where we were able to remove piezo2 from mice and test what happened. So when we induced osteoarthritis in our control mice in the black squares, you can see that they start to change their behavior. So they start to respond to lower forces compared to baseline. But the mice where we deleted piezo2, the ion channel never developed this kind of pain behavior. And a similar pattern was observed when we looked to see if the mice were putting the same amount of weight on their leg with osteoarthritis compared to the control leg. The control animals in the black squares start to put less weight on their OA leg over time, whereas the blue circles, the mice that are missing piezo2, never develop this behavior. So presumably they never feel pain, so they keep putting the same amount of force on their leg with osteoarthritis. So we went on to consider what is happening in osteoarthritis that makes the piezo channel act this way. And so there's different chemicals that are released in your knee joint over time in osteoarthritis, one of which nerve growth factor has received some attention in the pharmaceutical industry. And so what these observations initially highlighted is that when they created a new drug that was an antibody against nerve growth factor, in early clinical trials, they saw that this did have benefit for patients. So the patients who received anti-nerve growth factor started walking more compared to the placebo controls. Unfortunately, these pharmaceutical trials eventually got stopped, and they didn't end up getting FDA approval because there was an adverse effect associated with this drug treatment. And so we would like to better understand how to still use this pathway because it does seem beneficial, but try to come up with other ways of targeting it, maybe through targeting piezo2 in order to still end up with a drug that can get approved by the FDA. So I'll go through this a little bit quickly, but basically to say that both mice and humans express this piezo2 channel on their neurons that can also respond to different chemicals like NGF in your knee joint. So it seems like it's relevant translationally. And when we started to test this concept in mice by just injecting nerve growth factor into their knee joint, this seemed enough to cause knee swelling and knee pain, but the mice where the piezo2 channel was deleted didn't have this same response. So they were protected from the ability of NGF to cause pain in their knee joint. So we think in the future, considering different intra-articular strategies might be helpful in terms of also avoiding some of the adverse effects that other drug trials have seen, and perhaps looking at different ion channels expressed by your nerve endings inside your knee joint might be a way to go in the future to find new drug targets. This is just a quick pilot experiment to show that if we indeed try this concept out and we inject a drug inside the knee joint itself to block the piezo2 activity over a short period of time, because it's just a free drug, we could see a relief of pain. So with that, I'd like to thank all the people involved in this project. I mentioned Anne-Marie Malfay, who also leads the group with me here at Rush, our collaborators at Northwestern, Frank Coe, who does the cool 3D imaging, our funding sources, and particularly the ANRF for inviting me here today. So thanks, everybody.
Thank you so much, Dr. Miller. I really appreciate that. That was very interesting to listen to your presentation. I'd like to remind everyone if you could put your questions into the Q&A, and we'll try to get through as many as we can. I've seen some really interesting questions come through. So Dr. Jeffries or Dr. Miller, if either one of you want to jump in on these, feel free to. Our first question is, older studies include injections and/or surgery to regenerate cartilage, thus avoiding knee hip replacement. Is there any progression for the use of these therapies?
I can start on this. So the answer is not really. And, whenever these approaches have been applied to mouse models and expanded in human studies to larger populations, especially when they've been well controlled with, appropriate placebos and stuff, they're not as good as they seem at first blush. So I think there may be something there for example, intra-articular stem cell injections or PRP or something like that, but we don't know what it is yet. And it doesn't appear that the way that we're doing it right now is actually all that beneficial. So there's a massive interest in the field in this, but we just aren't yet there with an understanding of how these might work, I think. So I wouldn't recommend them necessarily for treatment.
Okay. Our next question is it common for obesity to develop after joint replacement surgery, even if it wasn't an issue before? Could this weight gain cause more symptoms to develop? Dr. Miller, do you want to answer that one?
I might hand that off to Matlock.
Okay.
That is a very intriguing question, and I don't know of any data on that. We often think of joint replacement as sort of restoring your functional ability and maybe associated with weight loss. I don't know if I've looked at studies to see what happens when there is weight. Obviously, there's a whole lot of pain, and there's a set of patients that don't respond to joint replacement, and they probably do get more weight gain. All of the other research that I'm aware of would indicate that basically anything that's associated with weight gain is likely to increase your pain sensitivity, at least as it is related to osteoarthritis. And a lot of that is from the inflammatory stuff that comes out of adipose tissue, of fat tissue. So I would expect it to make more symptoms, develop more symptoms if that were to occur. And the other kind of corollary to that to keep in mind is joint replacement surgery is not a panacea. Most people do very well, but there's still 5 to 10% of folks that undergo knee joint replacement that have persistent pain afterwards. And this is why the research that folks like Dr. Miller is doing is so important, because, why do you still have pain when there's no more biological tissue within that knee joint? And certainly there's something further upstream happening, right?
Our next question is, how long would you have to take a probiotic or focus on improvements to see any impact on your gut microbiome and osteoarthritis? And should I talk to my rheumatologist or a dietitian for help?
So I'll jump on this one since I was on the microbiome train a minute ago. The answer is we don't know, because the probiotics that humans take are not actually all that well studied. In the mouse studies, we are supplying this massive dose of bacteria, similar to what these microbiome transplantations that people undergo that have really severe infections, similar to that effect. So I would guess that in the stronger probiotics, like the yogurty drinks and stuff like that, that's been studied in knee osteoarthritis to some extent, probably within 3 months or so, you would expect to see some sort of effect. But we don't know for sure. And you can talk to your rheumatologist, although, some rheumatologists have read these studies and some haven't. So there's a decent amount of data about the microbiome in other autoimmune diseases, not just osteoarthritis, and dietary changes, particularly in Mediterranean diet, modifying the microbiome and resulting in improvements in disease. So I don't think it's just associated with OA. And I think there's a little bit more of a general understanding of dietary changes related to arthritis improvement.
I think that's very helpful for a lot of patients. Are there any new blood tests, imaging techniques, or other biomarkers that can help predict the progression of OA? Could these tools also guide us in choosing the best treatments?
So I could start and talk on the pain side of things. So the one still under research, so it's not yet ready for clinical application necessarily, but trying to kind of assay the status of your nervous system to kind of give you a sense of where you're at, I think is a promising area that could be utilized in like seeing just your general practitioner in the future. So the device that I described where they're applying forces to different parts of your body, they could kind of do baseline measurements, like you would get baseline blood tests and then see if you're starting to get changes and perhaps suggest ways of modifying your lifestyle that could slow that process down.
Do you have anything to add?
Yeah, there's a lot of interest in the blood-based biomarker field for osteoarthritis, and we're involved in that a bit because the other half of my lab that doesn't work on the microbiome studies something called epigenetics, which is the way that sort of the environment interacts with your genes. And we've published some blood-based epigenetic biomarkers that can be useful to predict if you're going to rapidly progress your OA or not. But those were developed based on data from these big osteoarthritis cohorts where patients are really well matched. So we don't really know how well that performs in the real world, right? But I think that we're moving in that direction. I think we're getting to where we'll be able to do some prediction in the future. And it's particularly relevant to the development of new drugs for osteoarthritis because, unfortunately, most knee osteoarthritis patients are going to take a long time to progress, and you can't really do a clinical trial that's going to last 20 years, So I think these predictive biomarkers will be important in making sure the right patients end up in clinical trials so we can actually see something in a few years,
Yeah. Where's a good place for people to find out about clinical trials?
I mean, you can always go to clinicaltrials.gov. Something that I will shamelessly plug, there's a new government agency called ARPA-H or ARPA for Health that was put into place by the Biden administration here a few years ago. And they're taking a different approach than the NIH, where they're focusing on individual diseases one at a time that are impactful. The first disease that they're focusing on is knee osteoarthritis. They have a big grant program. There were 5 research groups in the country that were selected to develop the first of the next generation treatments for knee osteoarthritis. And none of them are to the point where they're doing clinical trials yet, but I would tell you to keep an eye on the ARPA-H website here in a couple of years when they start coming up with the clinical trials for those interventions, because I think that'll be our best shot at getting, really cutting edge treatments for knee osteoarthritis, at least.
Yeah, that's incredible. I've always been a big, always trying to check out new clinical trials just when I run out of treatment options. It's kind of where I look first to see what's kind of on the cutting edge. So I think that's a great resource to have. Our next question is, having had joint replacements with others needed, how can I combat the development of OA? Dr. Miller, do you want to jump in?
Sure. So I would discuss this with your rheumatologist, but large-scale studies have demonstrated that lifestyle changes can be effective in slowing further progression. So we don't necessarily have any drugs that can be disease modifying at this point, but certainly regular exercise is recommended to improve symptoms associated with the disease. Keeping your body weight under control is also useful for both slowing the development and also keeping symptoms under control. I don't know if Matlock would have any other suggestions.
Yeah, I agree with that. The problem is we don't have any of these longitudinal studies, right? So we don't have a huge group of adults that don't have osteoarthritis that we followed for 20 years and tried to figure out the ones that did get it, what was different about them than the ones that didn't get it, right? So as best we know, weight management healthy diet, which coincidentally probably makes you have a healthy microbiome, right? Mediterranean style diet and exercise are probably the best we can do because we can't do much about our genetics and us slowly getting older. So
I feel like it would be great if rheumatology offices actually had a nutritionist on hand for
yeah.
their patients. It'd probably help a lot more patients actually see some more results in following up with someone regularly on top of their rheumatologist.
Yeah, I agree. It
Our
would be nice.
might suggest that to mine. The next question is about hormones. So can hormones make OA worse? With erosive OA, symptoms can come fast and furious along with menopause. 10 years later, and I've been losing use of two small joints and they're completely fused. Are there any studies on hormones in OA?
We have a decent number of mouse studies that would indicate that hormones play a pretty decent role in OA. And I mean, there's this massive amount of human data that show there's a spike in osteoarthritis risk in multiple joints in women around the age of menopause. So I think the answer is yeah.
That would make a lot of sense.
I don't know if Dr. Miller has additional stuff to add. I don't know the pain literature in animal models related to hormone, but
It's strongly correlated with the joint damage. So I'd agree with Dr. Jeffries' answer. I think that's a large contributor, especially up into a certain age. The male and female propensity to develop osteoarthritis is pretty similar, but it does seem to sharply take off in females after the age of menopause compared to aging males.
Okay. Our next question is how might new treatments target inflammation in OA? And are there specific markers that could help determine which patients will benefit?
Yeah, I mean, there's a variety of ways to look at this, and people have been working on anti-inflammatory type drugs in OA for forever. Ever since we started getting these fancy biologic drugs for rheumatoid arthritis that target specific parts of the immune system, people have been trying to apply those to osteoarthritis. And to be honest with you, they've mostly failed, even the ones that we would think would work really well. There's some interesting newer data that's being followed up on now that targeting certain parts of the immune system for a long time might reduce the progression in OA, but we just don't really know. I will say conversely, there have been a couple of kind of interesting clinical studies in patients that have knee osteoarthritis that if you give them one of the more generic older rheumatoid medications like methotrexate, it might improve their pain over time. And we know that low-dose steroids, which we hate as rheumatologists, we hate using long-term low-dose steroids, but they do kind of work in patients that have really inflammatory osteoarthritis, but we tend to stay away from them. Obviously, there's stuff that works, and I'm sure that we'll figure it out in the future, but we're just not quite there yet. I don't know if Dr. Miller has anything to add to that.
Yeah, I would say there's anecdotal evidence that it's probably beneficial, but again, trying to figure out the right patient population to plan a strong clinical trial, I think is what's been problematic. So it's kind of circling back to some of the same issues. If we could come up with tests to try to kind of get the right patient population enrolled for some of the clinical trials, you might see stronger effects for some of these anti-inflammatory drugs.
Yeah, and this is something a lot like, honestly, osteoarthritis is a lot like rheumatoid arthritis clinically in this way, because OA is probably 3 or 4 different diseases that we right now call all osteoarthritis, just like rheumatoid is probably 3 or 4 different diseases, right? And the clinical studies are all just treating sort of general OA with these interventions, but there probably is a subgroup of patients that respond really nicely and everybody else doesn't. So we've got to try to figure out who are those patients that'll respond to any given drug.
Interesting. And this is a question that someone else brought up, but I also have been interested in, and I thought I've heard some studies on it. Are there any studies on semaglutides to help reduce the development of OA?
Yeah, I think this is a really intriguing area. So there was a pretty large study that came out a couple years ago now where they followed 50,000 people. This was a study in China where a large portion of people started taking these anti-obesity drugs like the GLP-1 type of drugs. And so there was an effect. The people lost weight and they also had less development of osteoarthritis. I think there's other researchers looking at different ways of using these kinds of drugs in the context of osteoarthritis. So maybe without giving it systemically like you would to have weight loss, maybe local administration could still help in terms of reducing inflammation.
Yeah,
Do you
I
have
agree.
anything?
I'm really intrigued. Like, you know, you would expect that something that induces significant weight loss in a patient would probably improve their OA pain to some extent, right? But I'm really interested in these reported kind of other anti-inflammatory effects of these drugs and if they might add something on top of the weight loss. So I think that's a really interesting area of research. And I'm sure the drug companies will be more than happy to throw a bunch of money into that. So we'll see if we see anything over the next few years.
Yeah, it'll be interesting to watch. All right, I have one last question for you guys, and then we will let everyone go. Are there new, less invasive nerve therapies or nerve blocking techniques being developed to manage pain for patients who have had OA for many years?
I could start on that. So one of the less invasive options is using kind of topical creams that have capsaicin in as one of the active ingredients. And so capsaicin basically causes your nerves to become less able to respond to other stimuli. So there are side effects. It does sometimes people don't like using the cream because it can burn when you're first applying it because it's the chili pepper chemical. But in terms of you're able to apply the lotion yourself, that's pretty non-invasive. A more invasive strategy that is still we're still learning more about, but is actively, you can go to the clinic and have these types of procedures done is cryoablation. And so that's where they actually go in, and it is more invasive in that they're doing things to actually remove the nerve endings that are innervating some of the tissues that are painful. So we don't really understand the long-term effects yet of these types of surgical techniques, but they are being done.
And I don't know what the data on this are, but I know that anecdotally, some of the orthopedists that we work with here do those sorts of interventions, and also something similar called a genicular nerve block, actually in patients that are about to undergo knee replacement. And at least anecdotally, they're finding it's easier for the patients to do the physical therapy and the recovery after knee replacement. So that's something else I think that we have to remember, is that the recovery from what we would consider the definitive treatment for knee OA, which is replacement, might actually be improved by some of these interventions too.
That's incredible. Well, thank you so much, you guys. I really appreciate your time and all you put into your presentations, and want to thank everyone for joining us. I'm going to pass it back over to Ashley from ANRF.
Hi all. As we wrap up, I just wanted to take a moment to thank you so much to our fantastic moderator, Staci Stringer. We so appreciate you being here and sharing your story. Also to our wonderful researchers for sharing their innovative work towards understanding osteoarthritis. You can see firsthand the impact ANRF has had in supporting these incredible individuals in their quest for a cure. I would like to once again thank our partner AbbVie for your support of this webinar. And lastly, thank you to everyone who joined us for this great event. If you joined this webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps. We will be sending our live attendees a survey after the event. Your survey helps us find direction for future webinars, so please share your comments with us so we can continue providing important and relevant content to you. Thank you all for coming, and please remember to follow us on social media as well to stay connected. Have a great day.